welcome to the Brain and Transcranial Photobiomodulation Virtual Summit. I'm Dr. Joe Dodaro, your host. My guest today is Dr. Francisco Gonzalez Lima. Just a little introduction to this man who truly needs no introduction. Professor Gonzalez Lima and his trainees are world leaders in brain research and the relationship between brain energy metabolism, memory, and neurobehavioral disorders. Dr. Gonzalez Lima holds the George I. Sanchez Centennial Professorship at the University of Texas at Austin, the first endowed position in the U.S. honored in honor of a Hispanic professor. He has been elected fellow founding chair of neuroscience, Distinguished Texas Scientist Award winner, and the American academic director of the Texas Academy of Science. He has been elected fellow and U.S. counselor of the International Behavioral Neuroscience Society. He has been a visiting scientist in Germany, England, Canada, and Spain. Has delivered over 120 invited lectures about his research around the world. His brain research has been funded for over 30 years. He's contributed to over 400 scientific publications in peer-reviewed journals, conference proceedings, chapters, and books. And 12 years ago, he and his lab were the first to show in vivo, in animals, the upregulation of cytochrome C oxidase in the retina and the brain with transcranial photobiomodulation. So very, very excited to have you here with us, participating in the summit. Thank you, Professor.
It's a pleasure to be here.
So we want to just get a little bit of a background, and you've spent a lifetime, 12 years of your life, and more than that, even to getting your PhD and everything like that. But how did you get started with your photobiomodulation brain research? What's the story?
Yeah, the story started when we have been trying to find ways to stimulate brain energy metabolism, because brain cells are so dependent on oxygen metabolism in order to function. We were particularly interested in finding ways to do that, and that was our initial motivation.
And so your research question, the thing that came through, you kind of kept walking down this line was could there be what was the research question though? Like, how can you modify brain metabolism, or what was it that you were exactly looking for?
We were more focused on that. Some of the graduate students that work in my lab, we were interested in finding out what was the primary deficit in the brains of persons with Alzheimer's disease, late onset dementia. And we did studies that we published in the year 2000 using brains post-mortem, but these were very fresh brains. We had to go to Sun City, Arizona, with retired people live, and literally wait for people to die, and then go to with the pathologists and collect their brains as soon as possible after death. And once we collected enough of these, we started processing all of these brains, and what we found was that the most significant change was an inhibition of cytochrome oxidase. The activity of the enzyme was inhibited, and this was correlated with the progression of the disease. And we found that these so-called plaques and tangles were not really fundamental to the disease. There was no correlations, for example, between the plaques and the areas of the brain that were affected. And so from there on, we were interested, can we find a way to stimulate the activity of cytochrome oxidase in the brain, if possible, non-invasively? And could these result in improved cognitive function? And this planted the seed, and it took about 8 years to find a way to do it using red to near infrared light stimulation.
That's very, very, very helpful. Very helpful. So, at some point we'll be able to have like an overview, and you could kind of talk about this. But let's take a peek at your slides and see what you're going to explain to us today. Augmentation of human cognitive brain functions with transcranial laser. That is just, I mean, I'm excited. I'm excited already. Go ahead.
As a way of introduction, I would like to emphasize that brain cells are highly dependent on oxygen metabolism. As you know, if we stop breathing, we cannot sustain that condition very long. One of the characteristics of the brain, if you think in terms of analogies, is as if the brain is plugging into the wall, getting electricity through the wall, and you plug it out, you disconnect it, and it stops working. In other words, the brain doesn't really have the ability to store energy. It doesn't have batteries. And so the oxygen is needed for the process of producing energy. So what the methodology that we have been developing, we refer to as transcranial infrared laser stimulation using 1064 nanometer wavelength. And our goal is to augment cerebrovascular oxygenation, augment the oxygenation of the brain so that energy can be produced, so that functions such as cognitive functions could be improved. And the main target of these in terms of molecular targets is the respiratory enzyme cytochrome oxidase. This is an enzyme inside mitochondria in what is called the electron transport chain. And is the enzyme responsible for reducing oxygen to water in a process that is linked to oxidative phosphorylation that produces the metabolic energy. Without this enzyme, there's no possibility to use oxygen to generate energy. For example, you may have heard about a classic poison like cyanide. Well, how does cyanide work? What cyanide does is inhibit the activity of cytochrome oxidase. As soon as cytochrome oxidase cannot reduce oxygen to water, the process of using oxygen to obtain energy is stopped, and essentially the brain is unplugged from its energy source, and we pass out and then die. So the idea was to find a safe and non-invasive way to stimulate cytochrome oxidase. And using this laser light with low power but high fluence, that is, there is a high power density that can be translated into high energy density, with a penetration that can reach the cerebral cortex. And this is why we chose the particular wavelength to be able to reach into the human cerebral cortex. And if we, to give you an overview about the technique that we're using in our lab, again, it's important that we use the 1064 nanometer, but the reason for this is primarily to be able to penetrate deeper. It is possible to use other wavelengths that will have different kinds of penetration. And we have made an effort to demonstrate that this particular wavelength actually is targeting cytochrome oxidase, and it produces this photooxidation effect. So over here on the left, figure 8, we have an image of the one of the laser devices that we use that is commercially available. And on B is my forehead here showing. We have an aiming red light that you can see there. Normally we have only 2 centimeters away This is just for the purpose of the picture. And the idea here is that we can target a region in the forehead, and the equivalent region in the brain that we develop here in a model, where the areas that appear orange is the one that receive enough of this light to have an effect. So this corresponds to the prefrontal cortex, in this case, the dorsolateral prefrontal cortex. And it's important to point out that the effect is due to the light and not to heat. So here is from work with collaboration with Dr. Wan and colleagues, published in Neurophotonics. And when we compare laser stimulation with thermal stimulation. So laser stimulation is in red and thermal is in green. And over here in the vertical axis is the concentration of oxidized cytochrome C. And as you can see, the thermal stimulation at exactly the same level of warming of the forehead doesn't really produce any change. In this case, we're stimulated for 8 minutes, this shaded area. It doesn't have any significant change, whereas cytochrome oxidase concentrations start going up. And in fact, when we stop the stimulation, they tend to remain activated for some period of time. So the idea here is that this is a photooxidation effect on the enzyme and not a thermal effect. There is a window where this is most effective. We chosen the high end of the window, that is the longest wavelength, around 1,000 nanometers. And the reason for this is that the longer the wavelength, if we see here on the right side, the more penetration there is of light. Short wavelengths penetrate less, long wavelength penetrate more. However, as they penetrate, the energy level is decreased. So it is important that we have the correct dosing to the target. And also the long pulses are more effective at penetrating even with the same power level than short pulses. This is what we use continuous stimulation, which we call continuous wave, as opposed to pulsation. And another factor that is relevant for the brain is that it penetrates more through the gray matter than the white matter. So the white matter of the cerebral cortex is like a natural barrier, and the light doesn't seem to go much deeper than that. If we go about the molecular effects, focusing on cytochrome C oxidase, what I have here on the left is like a caricature showing the respiratory chain inside mitochondria. And these colored structures here are proteins, and the one that we're focusing is the cytochrome oxidase, which is the last one in this electron transport chain. When the light is provided, it is absorbed by cytochrome oxidase. And what this does, if we look inside the mitochondria here in terms of the biochemical process, we are photooxidizing cytochrome oxidase. So the photons are being absorbed, and this leads to oxidation of the enzyme, what I call here number 2. And the third thing that is happening while this process is going on is that protons, that is hydrogen that is ionized, is moving away from cytochrome oxidase. It's a proton pumper. And nitric oxide, NO, is also released from the enzyme, which sometimes it occupies the catalytic site of the enzyme. And the enzyme is able to reduce oxygen to water. When that process happens, ATP is synthesized. And as a result of this phenomenon and the release of NO, the capillaries that are around neurons become enlarged and increase the flow of oxygenated blood. So we have more oxygen bound to hemoglobin that is transported. It's important to point out that the hemoglobin is the one that transports oxygen, but the ones that receives the oxygen, over 95% of the oxygen that we breathe is cytochrome oxidase. So cytochrome oxidase is the one that catches that oxygen, and it allows them to do the energy production.
I just want to point out that this is an excellent description, and I want to make sure I have this correct, because you've made many good points here, that the oxygen is going to be absorbed by the cytochrome C oxidase. That's what you're actually stimulating, if I'm following you correctly, the absorption of oxygen, and then it's going to be help with the energy, but also the red blood cells within the blood vessels are taking up more of that oxygen also? Or is it just being, because of the vasodilation, you have more of them getting there?
Yeah, essentially the oxygen is transported in the hemoglobin, in these red blood cells, but the objective of those cells is to think of the hemoglobin like a pitcher in baseball, and the catcher of the ball is cytochrome oxidase. So the pitcher throws the oxygen to cytochrome oxygens, and essentially, in order for cytochrome oxygen to accept that oxygen, it has to have in what is called the oxidized state, the state which is being removed from electrons, and so it has more affinity for oxygen. Okay. And what the light is doing by photooxidizing cytochrome oxidase is increasing its affinity for oxygen, capturing oxygen, so that we increase the oxygenation, we increase the respiratory rate at the level of the mitochondria. And this is something that we demonstrated, we'll see directly by doing non-invasive measures. First, we did it invasively in animals, and even before that, we did it in brain homogenates, where we could directly measure the oxygen consumption rates and the activity of cytochrome oxidase, the enzymatic activity. And then we did it histochemically in animals. And then we follow up after a number of years to be able to do that non-invasively in humans.
So just to be clear, that was an excellent explanation for me. But so we're going to have we're going to have increased ATP synthesis within that mitochondria, which is going to be all that positivity. But also is the release of nitric oxide within the mitochondria or within the blood vessel? I mean, where is that? I know some of it happens in the mitochondria, but it's also transferred to an area to allow that increase in blood flow. So could you walk me through that?
Yeah, let me explain. Nitric oxide is a gas, just like oxygen. So when very similar, instead of two oxygen atoms, it has one oxygen atom and one nitrogen atom. So it can occupy the same catalytic pocket where oxygen occupies. So in order for the enzyme to work, it has to remove any nitric oxides from it. But the way cytochrome oxidase works in the mitochondria is, this is something that is a little misunderstood by some people in this field of photobiomodulation. The nitric oxide effect is not a sustained effect. When it releases the nitric oxide from the catalytic pocket, it's only a transient effect that gas will produce vasodilation of nearby capillaries. And however, that's not sustainable for the effect of the light. So what is happening is when the capillaries increase in diameter, they become dilated, more oxygenated blood comes in, and this oxygenated blood donates the oxygen to cytochrome oxidase, who reduces the oxygen to water. And the more cytochrome oxidase reduces the oxygen to water, locally, there is less oxygen available. So how do we get back the oxygen to be delivered to the local area? So the same enzyme, cytochrome oxidase, when there are low oxygen levels, now it switches from catalyzing the reaction of forming water with oxygen and hydrogen to now use nitrates and catalyzes the reaction to form nitric oxide. So when the oxygen levels go down, cytochrome oxidase becomes NO synthetase, releases more nitric oxides, which in turn vasodilates the blood vessels, brings back more oxygenated blood, and then the enzyme switches to use the oxygen for the process of generating ATP, oxidative phosphorylation. And this process, this cycle, needs to be maintained to have a sustained response. So just simply releasing NO from the catalytic pocket at the very beginning is not enough. You have to bring the oxygenated blood. The enzyme has to be able to use that oxygen to generate energy. But then when that oxygen goes down, the process repeats itself. The enzyme synthesizes or catalyzes the synthesis of more NO gas, and you again bring more oxygen to the...
Thank you for that clarification. I'm sure we'll look at that. We'll review that as we go forward. Thank you for that.
And let me show you now what I call the central thesis. So essentially, we want to augment this cerebral cytochrome oxidase, but we want to augment this oxidized form so that we can then facilitate the cerebral vascular oxygenation and eventually improve neurocognitive functions. So if you don't remember anything from this talk, I'd like you to remember what I bold-faced here. The infrared photons are actually absorbed by cytochrome oxidase. They photo-oxidize. Remember, cytochrome oxidase is an enzyme that is specialized to accept electrons. But in this case, it's accepting photons. Photons are like electrons, but they actually have no mass. So, you know, electrons have very little mass, so photons don't have any. So these photons are able to oxidize, called photooxidation, the enzyme. And when this happens, then that process of oxygenation that I explained takes place. And when more oxygen is available for more energy production, then we can enhance cognitive functions when we challenge the brain, or it can also serve as neuroprotection. In animal studies, we've shown how if we give toxins that affect the brain, the transcranial photobiomodulation can be protective.
That's a good thing.
Yes. So what I'd like to show you now is how do we measure whether this is really happening in the brains of humans. And I would like to say that no other group other than our collaborators and us have actually measured the effects of photobiomodulation, any type of photobiomodulation, directly in vivo in humans, and demonstrated that it upregulates cytochrome oxidase. And this is the device that we use. We adapted an ophthalmology chair, and we call this device broadband near-infrared spectroscopy. This is a custom-made device in our lab, and what we are able to do with this, if you look at this is one of the the model here is one of my visiting students from Rome, Italy, the University of Rome, and she has a very small head, so we choose her to give you an idea of the range. What I'll show you here is this is a custom-made laser device that we have in the lab, similar to the commercial one that we show you, and it's aimed at the forehead. And over here we have broadband light sources, and the idea here is that we apply broadband light to the head, and we have these sensors that are the smaller cables, and the smaller cables are going to be able to pick up, we call the detectors, photo detectors, which light was not absorbed through the tissues. And by knowing which light is not absorbed and which one is absorbed, by connecting this with the spectrophotometers, we can infer the composition of the chemicals in the brain. And we use these to infer the amount of cytochrome oxidase and the amount of hemoglobin. And importantly, to know whether the cytochrome oxidase is oxidized and whether the hemoglobin is oxygenated. So this photo was taken by Patrick O'Connor, a graduate student in my lab, who collected the data that I will show you in the next slides. All the data I'm showing you here has been collected in my lab. This is a graph of the concentration of cytochrome C oxidase, the oxidized form, and the shaded area indicates the 8 minutes of stimulation with the laser light. And we have two groups. There were 48 subjects randomly assigned to either sham stimulation, the same setup that you saw, but the effective light is not presented. In this case, actually, we use very low power aiming light. And the subjects have a protective eyewear. And then the other group, the solid green here, is the ones that receive the actual laser. And as you can see, during the 8 minutes, the amount of cytochrome oxidase that is oxidized is increasing in the presence of the laser, but not so in the sham condition. And after we stop, some of this continues. Usually by 5 minutes, it hasn't gone back to baseline levels. If we now look at what happens with the oxygenated hemoglobin, the oxygenated hemoglobin's response follows the cytochrome oxidase response. As I explained to you, after the cytochrome oxidase releases NO, the oxygenated blood starts bringing oxygen to the tissues. So this usually happens, as you can see, the effects here are happening after dose of cytochrome oxidase, and after we stop 8 minutes, the stimulation, it continues. We have only have a few subjects that we have maintained for up to 20 minutes because they have to be still in the chair and is still upregulated. We still have an increase in oxygenation to the brain. But it's important to point out that this is the oxygenated hemoglobin. If we look at the other kind of hemoglobin, the deoxygenated one, the one that is not carrying oxygen, there is no difference with the sham. If anything, there is a small trend to decrease the levels of deoxygenated hemoglobin. So what this means is that we're not simply increasing blood flow, because you can increase blood flow and blood volume in the tissue by just stopping your breathing. If you just stop breathing, there is more increased arterial pressure and blood that goes to your head. but that doesn't mean that you are going to get sustained oxygenation.
Wow.
So the key here is the increase in oxygenated hemoglobin, not just an increase in blood flow, which is, again, one of the issues that is not well understood in the photobiomodulation literature. And there is no other technique available where you can measure this directly in humans, as opposed to what we show you there. However, we wanted to know if increasing cytochrome oxidase activity, increasing oxygenated hemoglobin will have an electrophysiological effect on the brain. And this graph is from our collaboration with Dr. Wan et al., published in 2017. We have replicated this now with more subjects in our lab. Again, this will have sham and subjects. What this graph represents is the difference between the laser and the shams. And what we have here on the left is the frequency of the waves, the brain waves that are referred to as the wave frequencies. And on the right side, we're just classifying a range of those frequencies as this, the different waveforms that we can see in a human electroencephalograph or EEG. And we have here below the stimulation in terms of minutes. And in this case, we stimulated for 11 minutes, is the red line over here. And after that is a light off period. And the most clear change has compared to the sham, there is a large increase in the alpha, what is referred to as power density, spectral power density. That is the waves that are around here between 10 and 13 hertz or cycles per second are facilitated in response to the, and at about 8 minutes or so is the peak activity that you see here. There are also increases in beta waves, especially these low beta waves, and smaller increases in gamma. On the other hand, theta and delta, which are waves that appear when you are asleep, they go down. So this stimulation puts you in a alert state where you are also relaxed, which has been known, especially this combination of alpha and beta, to facilitate cognitive processes such as learning and memory.
Beautiful, beautiful. Now, so I guess the question that comes to my mind is, this is an induced state that is, say, are you allowing the cytochrome C to produce more energy to produce these states, or are you producing these states and you have the ability to sustain it with the increased ATP?
Yeah, this is a good question. The only reason this methodology is useful is because cytochrome oxidase is an inducible enzyme. What this means is that once you trigger it by this photooxidation, as time goes by, you come, for example, in animals, we can take their brains out. 1 day later, 24 hours later, measure cytochrome oxidase activity using quantitative enzyme histochemistry with a technique that I developed in the 1990s. And when we do that, we see that the enzyme is upregulated. Let me give you an analogy. This enzyme is responsible for oxygen consumption, aerobic metabolism, like I mentioned before. Let me go back to that slide. I illustrated here, you see how I illustrated how we have now more molecules of the enzyme that have formed. This is the long-lasting effect of the light. So I classify the effects of the light in two ways. I call direct effects when the light is on, what's happening, and then the indirect effect or secondary effects when the light is off. These enzymes and all of these cytochrome enzymes are inducible. What that means is that once you trigger them, there is a cascade in which they become upregulated, and this lasts hours or days. In animal studies, we can measure this. In humans. We don't have a direct way to measure this. But to give you an example, there is another type of cytochrome system that is in the liver that is for detoxification. So if you, for example, drink alcohol one day, the next day when you trigger that enzyme to detoxify the alcohol by breaking it down. So the next day, if you drink the same amount of alcohol, now you have more of this enzyme and it will be detoxified faster. So this is exactly what happens with cytochrome oxidase in the case of energy production. The enzyme is induced, and that's why at a later time, you can now produce more energy. Another analogy will be cytochrome oxidase itself in muscle, like aerobic muscle, you know, like the red muscle fibers that use oxygen primarily. When you work out to the point that no more oxygen can be used to produce energy is when you feel that you're out of air. And the reason for this is not that there is not enough oxygen. There isn't plenty of oxygen in the air. And you can take it all in, but you already exceeded the amount of cytochrome oxidase available. So what happens after that? Again, the next day you come and do the same workout, and now you have upregulated the amount of cytochrome oxidase in your muscle cells, and they can do more work in terms of obtaining energy, so you last longer. And the more you repeat these over time, the more aerobic capacity the muscle develops. The same thing we are doing with the laser. We're stimulating the aerobic capacity of the brain cells by facilitating the upregulation of cytochrome oxidase. Just like in the case of exercise, if you stop challenging the enzyme with more demand for energy, the enzyme will go back, will downregulate back to baseline levels. So presumably in photobiomodulation, we're going to have to repeat treatments at different periods of time to maintain the enzyme upregulated. And in my opinion, this is the biggest challenge for human applications, to find the appropriate protocols of when to repeat the photobiomodulation so that we can maintain this induced enzymatic activity and oxygen consumption levels.
Wow, that was perfect, perfect. And this would be one of the reasons why we have this neuroprotective effect, because you don't have to put the laser on and hurt the cell. You put the laser on and then 2 days later hurt the cell, or 2 weeks later hurt the cell, and it can resist. So thank you for that, for going a little deeper.
Exactly right. And in fact, we tested this in animals. We tested it by giving them toxin. We use rotenone that inhibits the electron transport. And the animals, for example, we injected that in the retina. The ganglion cells in the retina degenerated, but the ones that were given the photobiomodulation, and this treatment, we did this study during a 2-week period. They show no sign of neurodegeneration. And that was part of the study that we published in 2008 in the Journal of Neuroscience that was very convincing to us that this was an effective treatment that was even capable of preventing neurodegeneration.
That was Rojas' study that I was really excited about when it came out and referenced it quite frequently. It was like, do you understand what this means? And people were like, no, I don't understand. This is what it means. You know, no, they didn't get it. So thank you for that.
So let me show you another way to look now what's happening throughout the brain, because even though we are targeting that prefrontal region, the electrophysiological activity happens throughout the brain. That prefrontal region is more like an orchestra director. We call in neurocognitive science that is top-bottom control. In other words, when you facilitate the energy capability of the boss of the brain, the prefrontal cortex, then it can orchestrate different areas. For example, this is using a technique called E-Loretta, focusing on this alpha band from 8 to 13 hertz, where we saw the increases in power density, and looking at some of these minutes. And remember, this is the right hemisphere where we're aiming the laser. This is the left. And at the beginning, it's all start happening on the right side in the front. And we see activity also in the posterior part of the brain. There are direct connections from these frontal to occipital regions that you get activated at the beginning. And as time goes by 8 minutes when we have the maximal response, then we have an entire system that becomes active. Not only that prefrontal region, but the posterior parietal regions and occipital regions, anterior and posterior temporal regions. And at this time, it starts happening on the other side of the brain. As time goes by, then you see the other hemisphere becoming engaged. And by the end of the stimulation, here 11 minutes, the two original sites are the ones that are maintaining this synchronization of alpha activity. What this means is that an entire network, and this network turns out, because we initiated the effects in the dorsolateral prefrontal cortex on the right side of the forehead, we are stimulating this frontal parieto occipital network that is usually the one that is engaged in cognitive processing. So by selecting to stimulate there, we are activating a network of regions that is involved in cognitive processing, and that explains why we can have a cognitive improvement.
So one of the things that in reviewing some of Wang's work and the stuff's coming out of your laboratory, I kind of describe it as like once that you activate a part of the brain, then it's like a pinball machine after there. And you demonstrating that it's not random, it's following pathways that we know about. And that's what you're going to elucidate on. Am I correct in my metaphor?
You are absolutely correct. And that means that you could stimulate, for example, instead of lateral locations, you could stimulate medial locations and activate another network preferentially. But what we have found, interestingly, is that right-sided stimulation always seems to work better than left-sided. And the reason for this is that the left prefrontal cortex here, especially this dorsolateral region, has more outputs that influence these networks than the left does. So the key is to stimulate a hub that is predominant in terms of outflows, and as opposed to a hub that is predominant in terms of inflows. And this is not something that I understood when I started this work. I thought I could stimulate any part of the brain and get similar effects. And it's not the case, because the brain is a network. It's not just a collection of isolated modular regions. And so if you stimulate that part of the network, that is where you have outflow to other regions, that's going to be more effective than if you stimulate a different part of the brain.
That's such an important clinical, I mean, it only takes 20 years to make that conclusion, right? It's an important clinical point, I believe.
Thank you. So let me show you now, I have preliminary data here in collaboration with Dr. Jorge Almeida from our medical school psychiatry department that I'm collaborating with him now, moving this technique to clinical populations. And this is another technique that is based on functional MRI, magnetic resonance imaging. In this particular technique is called the ASL, or arterial spin labeling. This is a technique that magnetizes the red blood cells, and you can follow them moving so you can measure cerebral blood flow throughout the entire brain. And again, we stimulated, this is the right hemisphere here. We stimulated in the right hemisphere. If I make a cross section here in the image, this functional MRI image, and show you a coronal vertical section, you can see this is the area that we're stimulating, and this is the increase in cerebral blood flow in that area as a result of the stimulation. This, let me tell you, this is not that we are stimulating right now. These individuals received the stimulation immediately before we did the MRI, the scans. And in this particular case, we were stimulating once a week for 5 weeks. And then what we see is that this area now is facilitated in terms of cerebral blood flow. If I look at what is called the horizontal or axial image, if we cut through here, you can see this is the region of the right prefrontal cortex where we have the cerebral blood flow increase. However, as you can see, it's not the only region. This is where the stimulation was delivered, but we have all the areas similar to what we saw with the electrophysiology. Now we're seeing here with the cerebral blood flow. Again, this is the first time this being demonstrated. And look, interestingly, all the areas, for example, here, this is what we call the posterior cingulate cortex. This is the area that shows the largest decreasing metabolism, especially in cytochrome oxidase activity in the brains of Alzheimer's patients that we discover when we look at the postmortem brains. So by stimulating the prefrontal cortex on the right side, we are increasing blood flow to this region that is far from where the light is reaching.
And the point is that is a bilateral activation that we're looking at. And if I'm not mistaken, the posterior cingulate gyrus is going to be into our emotional. This is an important emotional area. So when we think that we're treating the prefrontal cortex for depression or these sort of things, we're having downflow, downwind, activation. And
correct. And it is, yeah, that's an important observation. The reason this is important for cognitive functions, for example, to memory, is that we preserve in memory events that have emotional content, emotional value, that are significant to the organism. Things that are unimportant, irrelevant, we don't store in memory. So in order to facilitate memory formation, you need to engage regions like this posterior cingulate cortex. And when this region becomes hypometabolic, like in the case of dementia, then you have an inability to form new memories. And so this is a wonderful thing for this technique, because we don't need to be able to directly stimulate there through, which is in a fairly inaccessible part of the brain for non-invasive stimulation. We only need to engage a network that taps into that region, like in the case of this right-sided stimulation that we use here.
I want to point out one more thing, because this is showing the adaptation of the brain. Because just to repeat, so I'm clear, you treated these people weeks before. How many times did they get treated before they did the scan?
5 times.
5 times. So that was daily for that week?
No, this was once a week. We still are trying to find out different schedules, like once a week, twice a week, 3 times a week. And see, definitely once a week, it has an impact on the cerebral blood flow. But we need to optimize these protocols. And in this particular study, we want to optimize this for individuals that have psychiatric disorders that are chronic that lead to cognitive impairment.
Wow.
So we want to be able to improve their cognitive impairment, to improve their cognition, so that they can then respond to their other treatments. So somebody, for example, who is depressed, you cannot do psychotherapy, talking, and these people are just can't think. So you need to, one of the most neglected areas in psychological and psychiatric research is the role of cognition. And you need to improve your thinking if you want to improve all the functions of the brain, because they are linked to each other. Just like what we talk about here about forming memories. Well, these are linked to your emotional system, because you want to store what is emotionally relevant, what is relevant to the organism.
Fabulous, fabulous. I had like 6 wows on this one slide. So.
So let me move to the next one. This is an example of some of the studies that we did. In this study, we target what is called executive functions. These are decision-making functions that are necessary for all of our daylight activities. But we published this several years ago in the Journal of Neuropsychology. And here we focus on stimulating the lateral part of the forehead. This is actually the forehead of my former student, Dr. Nate Blanco, that led this study. And we use the Wisconsin car sorting task. Why we did this? Because this is regarded as the gold standard for neurophysiological measure of executive function. So if you talk to neuropsychologists and ask them, is there any technique that can measure this prefrontal function? Which one will be the one of choice? And this is the one that they will agree on. And sure enough, I can tell you that they told me, oh, there's no way you're going to be able to improve that by just shining light in the forehead. And much less to our group of young college students that already had high cognitive executive functions. We went ahead and did the study anyway. And even with the small sample size on 15 in the placebo group and 15 in the laser group, we found significant differences. It is important to have a placebo group. In all of our studies, we have placebo or sham, that is fake stimulation. And if you look at the placebo and the active, you can see that the active has a greater proportion of correct responses. And if we look in detail what is happening in this technique, you change the rules that are used to make the decisions. So for example, in one case, the rule may be the colors, in others, maybe the numbers. And people, you don't tell them the rule, they have to figure it out. So what happens usually is that they are making a number of trials to reach a criterion to move on. But when you change the rule, then they have to make more trials to figure out what the rule is. Well, when the ones in the placebo group did that, like normally happens, however, the ones in the laser group, they didn't have to increase the number of trials to figure out the new rules. They could understand what they adapt their executive functioning to the new change nearly twice as
They
better
would.
than the other ones in terms of, less the number of trials to reach the criterion. So this is an example of the techniques that we've done.
What did they say about, excuse me for 1 second, what did they say when you tell your psychologist friends or colleagues, look at what we did? You talked about cognition as far as performance learning, but figuring things out.
Correct.
What do they say when you show them results like this?
Well, at the beginning, they predicted this would be impossible. Young, healthy people, and in this particular case, this was only one session of 8 minutes of stimulation that was done before they were challenged with this executive task. And well, they predicted that this could not happen. Well, if you improve oxygenation to the brain and energy resources, the brain can operate at a higher efficiency level. And then you can make less mistakes. Just like if you know that if you're tired, you haven't eaten and you don't have enough, produce enough energy resources. By the way the food that we eat leads to energy in the brain is that what we eat, it becomes converted into electron donors that go in that electron transport that I just explained at the beginning. And when you add a phosphate to form ATP, that phosphate, when you break it out, releases heat, and that's what we call calorie. So the amount of food that you eat becomes electron donors, and then that electron donors result in phosphorylated ATP that many reactions in chemistry, when you break something, they release heat. It's called exothermic reaction, and that's what the body uses for energy. We use heat. So we bypass the whole system by donating photons to the electron transport chain and produce energy without the use of food resources.
Wow, beautiful, well stated.
And let me show you another study. This is the most recent one that published with Holmes. Holmes was one of our students that did their honors thesis on this project. Here we have two other techniques. This one that we call the psychomotor vigilant task. That is a task for sustained attention. We have actually years before demonstrated that we can improve this with transcranial photobiomodulation. And we had a delay match to sample memory task. This is a so-called working memory task. So in this study, actually, we replicated these findings. This is a correct score from before and after the task in individuals who received the laser. And are the same task in the sham group from before and after. There's absolutely no difference in their performance. So this was a replication that these effects are not due to practice. If you give it before and after, they're due to an improvement of the correct responses in the task. What was important of this study that we added was monitoring what was happening in the brain in terms, for example, of the oxygenated hemoglobin. And these are millimoles, molar amount of change of oxygenated hemoglobin. And PBT refers to this sustained attention task, and the DMS to the working memory task. And this is before the laser stimulation. You can see that there are some increases in the amount of oxygenated blood that is used by the brain to solve the task. But then we provide the 8 minutes of laser stimulation. We didn't record during this time because the laser itself may affect the monitoring device, the detectors. then we perform the same task. And now the brain is performing the same task at a better level, but as you can see, is using more than twice the level of oxygenated oxygenation to this prefrontal region where we did the monitoring. So the reason we are improving performance is that now the brain can, we facilitate the process of oxygenation to the brain and energy production. And this is the reason the performance improves. And there is a relationship between how much the brain gets oxygenated and how well they do in the task, which is something that we are planning to use for screening individuals when we go to clinical populations to first determine if physiologically they respond to our treatment and what is the optimal dosing, so that we can use that to decide on the protocols.
Very elegant research.
And in this last study, it was a small study, only a dozen people. This is what we call a preliminary or pilot study. These were older people from 49 to 90 years old. And the point here was to show that this population can also benefit. We use the same task as before with the young individuals, the psychomotor and the working memory task. And again, these were given 5 weeks of once a week stimulation. And what we see here is that in the sustained attention task where you measure the reaction time, so it is better to have a smaller reaction time, be able to make the response faster. And as you can see, acute after one session or after 5 sessions, weekly sessions, you can reduce your reaction time in this attention task. Again, this was something that colleagues in psychophysiology, they say, you cannot change the reaction time of somebody just by exposing them to a light for a few minutes. And you can, yes, you can. People can, if you have improved oxygenation and energy resources, you can improve all of these brain functions. And again, you make less laxes when you're doing this, because you have to make a response quickly within half a second, and you make less of these mistakes. And in the memory task, you also improve the number of corrects. However, in this task, it was too easy, so we reached a ceiling effect where people could not improve more than the majority of the correct answers. So this was also published. And so this leads me to the conclusion that transcranial infrared laser stimulation can be used non-invasively in a way that is efficacious to increase cerebral cytochrome oxidase and oxygen metabolism and support neurocognitive functions in humans. It is safe, it's innovative, it can be done in healthy humans, and we hope to move these to people that require, I refer to as neuropsychological rehabilitation, but this could be a number of different conditions. Potential applications, these are the first one that we are testing now. Aging-related cognitive decline that happens in all of us after 45 years of age. We have a measurable decline. And attention and memory trauma, ischemia, when there's less blood flow going into the brain with less oxygenation. And other psychological and neurological disorders, as I mentioned, all psychological psychiatric conditions, when they become chronic, they compromise also cognitive function. And this is part of the reason they cannot get appropriate relief from their conditions. And I'd like to give credit to, I selected some of our publications from 2012 to last year that I included here. And that gives, we have a collaborative group in Texas. Dr. Liu is a bioengineer that has been fundamental in developing the methodology to be able to make these measurements in the brain. Some of these are my graduate students, like Patrick O'Connor, that you saw the data that I presented, Celeste Saucedo, that produced data for this executive function. My lab manager is Dr. Barrett, and he's the second author here, and this was the honor student that did that project. And there have been a number of dissertations, and many of these people have obtained their PhD degrees doing these particular projects in our labs. So I leave it there.
I think that you touched on this by talking about the future when we thank you so much for your time, Doc. But I also want to talk about the future, and I have a little bit more gray hair than you, and we want to see this research go further. And you have had that great influence by growing the field of research, by growing more researchers that are still looking at this. Would you agree with that? Is that part of the future?
I hope so. My main concern is that we need to move and do this kind of control studies. Control studies where you have sham placebo groups where you can be certain that the effects are due to our interventions. And it was fine at the beginning to have all these, few cases, observation, anecdotal results. But if this is going to become a mainstream scientific intervention, we have to be rigorous and we have to have control studies. And it is unfortunate that some of people in this field have not implemented that approach yet. They continue to make a lot of claims with very anecdotal data, and this is not going to help anybody. And this technique is only as good as the particular protocol and intervention that we make. It's not as if any type of stimulation is going to work for any condition. So it is our challenge to be able to investigate this, and unfortunately it's slow. Right now we are in a period where we are not able to run more participants in these studies.
Do the trials. I know that's a limiting factor on right now with all the researchers that we're going with. So you've done a great job reviewing the last 20 years of your life. Now, we went down to the microscopic level and the cellular level. I want to ask you to turn your lens to the future. Mm-hmm. Tell me, what do you think the future is? Let's just go to 2030 and talk to me about the 2030 Brain and Transcranial Photobiomodulation Virtual Summit. What do you think that we will see in the future, 10 years from now?
I think we will see what has happened in technology over the last decades. We will see portable, miniaturized versions of what we're doing instead of that chair that I show you there with all this. We will have portable devices that will be able to monitor the effects of the stimulation. And we'll be able to have also stimulation delivered by those smaller portable devices. I do hope that we can do this instead of with lasers that present risk for the eye, for this, that we could do this using LEDs, light emitting diodes. These diodes are getting better at narrowing their particular frequency range so that we can produce similar to lasers that have only one frequency. And it is, I hope that this is going to happen. And Right now, many of these LEDs are not suitable. They are inefficient, and that means, and you can tell that very easily when electrical power, when it's converted to light, if some of it is not converted to light, it becomes heat. So if you actually touch your LEDs and you feel the heat in the device, that means a very inefficient device. A lot of the electrical power is becoming heat, releases heat. This doesn't happen with these lasers that we use. They're pretty cold lasers. They are high efficiency. So I believe this is going to happen. We'll have LEDs that will perform very similarly. We're not still there yet, but I see that as happening in the future. And in the future, I will see that people who have a particular clinical condition then could be tested whether they're responsive to the stimulation. You could adjust the dosing for the particular individual by doing a dose response using, for example, the oxygenated hemoglobin could be a response in the brain, and then set that dose for the subsequent treatments. And they could have these treatments themselves at home. And you could also monitor their performance just like what we're doing now at a distance. They could, by getting into the phones, they could provide data to you, data from the recordings of their physiological responses, but also responding to questions about how the treatment is performing for them.
I agree. Thank you so much for your insights, your time, and all the work you and your team have been doing to help us get a better understanding of the science of this and the evidence of this great technology. Thank you, Doctor.
Thank you very much, Dr. Di Duro.